Hello everyone, welcome to another lecture on the Swayam course Microbial Technology. Today we will be discussing on the topic retroviruses. Myself Dr. RGB, working as an assistant professor in the Department of Biomedical and Bioinformatics UTD CSVTU Bhilai. So what are these retroviruses? Retroviruses are a type of virus that carries their genetic material in the form of RNA rather than DNA. What makes them unique is their ability to convert their RNA into DNA after they infect a particular host cell. This conversion process is done by the enzyme called as reverse transcriptase. Now, here are some basic characteristics about the retroviruses. First one, RNA based genetic material. Unlike most of the virus that uses DNA as their genetic blueprint, retroviruses uses RNA. This RNA is a single stranded, meaning it consists of just one chain of genetic material. It also has the enzyme called as reverse transcriptase. So, after entering a particular host cell, a retrovirus uses reverse transcriptase to turn the RNA into DNA. This DNA gets integrated into the host cell's own DNA, becoming a permanent part of the cell's genetic material. Now, they also tends to form the provirus. Once integrated, these DNA called as provirus. This provirus can stay hidden within the host DNA for a very long period of time, sometimes for the life of the cell, and can be passed on when the cells divide. Imagine a retrovirus like a spy who can sneak into a building, changes its identity and then blends into the crowd. The spy can remain hidden and later can cause the trouble. So, let us also discuss some of the historical perspective of retroviruses. So, retroviruses were first discovered in early 20th century. In 1908, Danish scientists Wilhelm and Olof Bang discovered that leukemia in chickens could be caused by a virus, which we know now known as retroviruses. This was one of the first clues that viruses could cause cancers. In 1950, discovery of reverse transcriptase. The enzyme reverse transcriptase was discovered in late 1950s by Howard Temin and David Baltimore, which was a groundbreaking findings. This enzyme allows retrovirus to convert RNA into DNA, a process that was previously not known in the field of biology. In 1980s, the HIV and AIDS epidemic occurred. In late 1980s, we saw the identification of HIV, that is immunodeficiency virus, a retrovirus that causes AIDS. The discovery was such a crucial in understanding of how retroviruses work and their impact on the human health. Now, this also in the next series, it was also awarded a Nobel Prize recognition. The discovery of reverse transcriptase and the understanding of retroviruses, its role in diseases like cancer and AIDS, has earned several scientists including Temin, Baltimore and French scientist Luc Montagnier. The Nobel Prize was shared amongst them. Now, let us proceed to learn about the components of retroviruses. So, the first component is their genetic material. Understanding of structure of retroviruses is crucial because it explains how these viruses function and how they can infect a particular host cell. Let us break down each component and aspect of their structure. So, in the first we will study about the genetic material. So, first one is single stranded RNA. So, retroviruses carries their genetic material in the form of RNA, not DNA. Specifically, they have single stranded RNA, also called as ssRNA, meaning their genetic material is composed of single chain of nucleotides. Retroviruses typically have two identical strands of RNA, which are packaged together inside a virus particle. This is somewhat unique among the viruses, as most of the other RNA viruses only carries one single strands of RNA. Let us study role of RNA in the infection. The RNA contains the information like what the virus needs to replicate and to produce more number of viruses once it infects the particular host cell. However, because the host cell primarily work with the DNA, the retroviruses must first convert in its RNA into DNA using the enzyme called as reverse transcriptase. Now, the next component is also called as capsid or the protein coat. So, the capsid is a protective protein shell that encapsulate the retrovirus's RNA. This structure is crucial because it shields the genetic material from the damage while the virus is outside a particular host cell. It helps to deliver the RNA into the host cell once infection begins. The capsid is made up of protein subunits called as capsomers, which comes together to form a strong, stable structure. In retroviruses, the capsid often has a cone shaped or a cylindrical form, which is different from the spherical or icosahedral shape seen in many other viruses. The capsid not only protects the RNA, but it also plays a key role in the initial stages of infection. It ensures that the viral RNase is safely delivered to the host cell cytoplasm, where the process of reverse transcription and integration into the host DNA will occur. Imagine the capsid as a suitcase that carries the virus precious cargo, and it safely delivers the virus particle or the virus RNA into the host cell until it reaches its destination, that is inside the particular host cell. Next important component is envelope. So, surrounding the capsid is a lipid membrane known as envelope. This envelope is derived from the host cell's own membrane, which the virus steals and it buds off from a cell after replication. This makes the envelope a mix of viral and host cell components. Embedded within the envelopes are the viral proteins such as glycoproteins, which are essential for viruses to recognize the binding sites in a particular host. These proteins act as keys that fit into specific locks, that is receptor on the surface of host cell, allowing the virus to enter the particular host cell. The envelope is crucial for the infection process. Without it, the virus would not be able to efficiently enter into the particular host cell. The envelope also helps the virus to invade the host immune system by resembling the host cell's own membrane, which makes it harder for the immune system to detect the virus. Now, we will study the very important protein that is called as reverse transcriptase. This enzyme is one of the most crucial components of a retrovirus. After the virus enters the host cell, the reverse transcriptase converts the viral RNA into DNA. The reverse transcription is necessary because the host cell's machinery works only with the DNA, not RNA. The high error rate of reverse transcriptase during the conversion process leads to mutation in the viral DNA. These mutations can result in genetic diversity within the virus population, making it harder for the host immune cells to fight off the infection and complicating the treatment process. Now, next enzyme is also called as integrase. So, once the viral RNA has been converted to DNA, the next step is for the DNA to integrate to the host cell's genome, The enzyme integrase is responsible for this particular process. It cuts the host DNA and insert the virus DNA into it, where it can remain indefinite amount of time. Now, the integration of viral DNA into the host genome means that the virus becomes a permanent part of a particular host cell. Every time the host cells divide, it copies the viral DNA along with its own DNA, spreading the information to the new cells. Next enzyme is protease. Protease is an enzyme that processes the virus protein once they are produced by the host cell. After the virus genetic material has been integrated and expressed by the particular host cell, proteases can cut long chains of viral proteins into the functional units. These processed proteins are then assembled together to form new virus particle. Without protease, the viral proteins would not be properly processed and the virus would not be able to assemble together correctly, making this enzyme very essential for the maturation and release of infection. Now, we will learn about the morphology, its shape and size about the retroviruses. So, retroviruses generally have a spherical or slightly irregular shape when viewed under the microscope. Now, retroviruses are relatively small, typically ranging from 100 to 200 nanometers in the diameter. Despite their small sizes, they pack in all the components necessary to hijack a particular host cell and then later they reproduce. Now, they also have layered structure. The retroviruses has almost 3 components. The first one is we have already studied about the genetic material, followed by the capsid, followed by the envelope. Now, let us talk about some of the unique features of these retroviruses. Retroviruses have some unique feature that sets them apart from all other viruses. Two of the most important features are reverse transcription activity and ability to integrate their genetic material into the host genome. Let us take a closer look of these processes. First one, reverse transcriptase. This enzyme is central how the retroviruses operate. Without it, they would not able to replicate inside the host cell, and also we have studied this in our previous topics. Now, the integration into the host genome. Now that virus RNA has been converted to DNA, the next critical step is for the retrovirus's life cycle is to integrate its DNA into the host genome. This step is what allows retroviruses to establish a long term infections. So, in the upcoming slides we will see about the mechanism process. The integration process is carried out by the enzyme called as integrase that we have studied in previous slides. After reverse transcription converts the viral RNA into DNA, integrase take these viral DNA and insert them inside the host cell. This means that virus DNA becomes a part of the particular host cell genetic material. Whenever the host cell replicate, it allows the replication of viral DNA as well, ensuring that virus can persist in the host cell for a very long amount of time, sometime even for a lifetime of the host cell. The integration of viral DNA into the host genome has several significant consequences. First, it allows the virus to hide from the immune system by blending in within the host own DNA. This makes it very difficult for the immune system to recognize and eliminate the viruses. Second, the integration of viral DNA known as proviruses can remain in a dormant phase for years, only reactivating to produce new viruses when they are triggered. This dormancy and reactivation makes the retroviral infection chronic and challenging to treat. Now, let us briefly talk about life cycle of retroviruses. Understanding the life cycle of retroviruses is essential to grasp how these viruses operates, replicate and causing the long term infection. Each step in the life cycle builds upon the previous one, leading to the production of new viruses that can go into infecting the other cells. Let us walk through each step, linking them together to show how the process unfolds. So, here are some steps in the life cycle. First one is attachment and entry. The life cycle of retrovirus begins when the virus comes in contact with a susceptible host cell. The first step is called as attachment and entry. The retroviruses recognize and binds to a specific receptor of the surface in the host cell. This binding is highly specific. Only cells with the right receptors can get infected by the virus cells. The viral envelope protein acts as key fitting into the host cell's receptor, that is a lock. Once the virus is attached, it must enter the host cell to begin the infection process. This typically occurs through the membrane fusion or the receptor mediated endocytosis. In membrane fusion, the virus envelope merges with the host cell's membrane, allowing the viral RNA and enzymes to enter the cytoplasm. Once inside the host cell, the next critical step would be retrovirus life cycle is reverse transcription, which we touched on earlier. This step converts the viral RNA into DNA. Inside the host cell cytoplasm, the enzyme reverse transcriptase goes to work. It converts the viral RNA into the complementary DNA or the cDNA. This DNA is then copied into double stranded form, making it ready for the integration into the host cell's genome. Reverse transcription is a very key step, and a virus cell must follow it specifically to integrate itself inside the host cell genome. So, with viral DNA now prepared, the next step in the life cycle is integration. This is where the viral DNA is inserted to host cell's genome, becoming a permanent part of host cell. The enzyme integrase carried out this particular process that we have also briefly touched in previous slides. The integration is defining the features of a retroviruses, enabling the virus to hide inside the host genome and invade the host cell's immune processes. Once integrated, the viral DNA can be activated to produce new viruses. This activation leads to the next step, that is transcription and translation. So, when the provirus is activated, the host cell's machinery transcribes the viral DNA into mRNA, just like it would with its own genes. This mRNA contains the instructions to produce viral proteins. Next step is translation. The mRNA is then translated by the host cell's ribosome into viral proteins. These proteins includes the structural components like the capsid and enzymes like reverse transcriptase, which are essential for assembling new viruses. Finally, the newly produced viral components must be assembled together to form a complete virus, which are then released from the host cells to infect other cells. First, we will look at the assembly process. The viral RNA and proteins come together in cytoplasm to form new virus particles. The RNA is packaged inside the capsid, and then envelope proteins are incorporated into the budding virion particles. Now, the next one is release. The new virus exits the host cell through a process called as budding. During budding, the virus take a piece of host cell's membrane within it, forming the viral envelope. This process does not immediately kill the host cell. It allows it to continue the producing new virus for some more amount of time. We will look into the process reverse transcription. Reverse transcription is a crucial process in the life cycle of retroviruses, as it allows the viruses to convert their RNA into DNA. It is a key step that we have seen previously. Now, we will closely look at the process of reverse transcriptation. To begin with, reverse transcription involves the conversion of viral RNA into DNA. So, which is necessary step for the virus to integrate its genetic material inside the host genome. Once the retrovirus enters inside the host cell, its RNA need to be converted into DNA, so that the host cell can use it. This conversion is carried out by this particular enzyme, which synthesizes the complementary DNA strands called as cDNA from the viral RNA template. The newly formed single stranded cDNA is then converted into double stranded DNA, making it a very stable and ready for integration into the host genome. Now, there are certain error rates associated with this particular process. One of the most significant characteristics of reverse transcription is its high error rate. This high error rate has important implication for the virus ability to adapt and for the challenges in treating in retrovirus infections. There are high mutations rate associated with it. Unlike cellular DNA polymerase, the reverse transcriptase lacks the proofreading ability. That means that when it copies the virus RNA into DNA, it often makes mistakes, also called as mutations. These errors accumulate in the viral genome, leading to the high mutation rate within the virus population. The frequent mutation can result into the virus quickly evolving the resistance towards the antiviral drug, as the new viral variant may no longer be susceptible for the treatment designated for the target or the particular protein. Additionally, these mutations can help the virus to invade the host cell's immune system, making it harder for the host cell to recognize and fight against the infections. Now, the next one is integration into the host genome. Integration into the host genome is defining the features of retroviruses, allowing them to establish a long term infection that can be difficult to detect and later on treat. This process, driven by the integrase enzyme, has significant implications for the persistence of viruses within the host cell. Let us explore how the integration happens and the long term consequences associated with integration. So, after the viral RNA has converted to DNA through reverse transcription, the next crucial step is the retrovirus's life cycle is to integrate these within the host cell's genome. The integrase enzyme is responsible for integration of viral DNA into the host cell's DNA. After reverse transcription process, the double stranded viral DNA integrase locates the suitable spot within the host genome and make cuts to the host DNA. The viral DNA is then inserted into these cuts and becoming the part of host cell. Now, the next process is transcription and translation. After the retrovirus has successfully integrated its DNA into the host genome, the next crucial steps are the transcription and translation, and we have already established the understanding of transcription and translation in our previous slides. Lastly, we will look at the assembly and release. The final stage of retroviruses life cycle involve assembling of newly produced viral components into the complete viral particle or the virion particle, and then release these virion particles from the cell. This process ensures that virus can spread and infect the new cells. First one is virus assembly, packaging of viral RNA and protein after the transcription and translation process. This is completely governed by the virus assembly. In the cytoplasm of the host cell, the viral RNA which was transcribed earlier is packaged together within the viral proteins. The RNA is enclosed within these capsid proteins, that is a particular cell, that is a particular protein shell that protects the genetic material. Now, the next process is formation of new virion particle. Once the viral RNA and proteins are properly packaged, the next step is to assemble these packages into virion particles. So, the capsid protein self assemble around the viral RNA and enzymes, forming a stable and protective structure. This newly formed capsid along with the other viral proteins can make up the core of virion particles. The virion envelope protein, which are embedded in the host cell membrane, will now incorporated into the virion particle during the process of budding. The formation of new virion is a self-organizing process, meaning that the viral components naturally come together to form a complete virus particle. Each virion is now fully assembled and ready to be released into the host cell. Next process is budding and release, that we have already seen in the previous slides. In this, there is one crucial stage of maturation of virion particles. So, once the virion particle is released, certain viral enzymes such as protease continue to process the viral proteins, finalizing the structure of virion particle. This step ensures that all the components are in correct configuration and that the virion particle is fully functional and capable to infect other cells. Without maturations, the virion particle will not be infectious even if all the components are present. The final step transforms the newly budded virion particle into a fully mature and infectious viral particle. Now, briefly we will look at the classification of retroviruses. Retroviruses can be classified into different groups based on their characteristics and types of diseases they cause. Understanding these classification helps us to see how diverse retroviruses are and how they can lead to different health problems. Let us explore these main classes. They are oncoviruses, lentiviruses and spuma viruses. First, we will look at the oncoviruses. Oncoviruses are a group of retroviruses that have ability to cause the cancer in their host cell. Let us look at how they do this with the help of some examples. So, oncoviruses are retroviruses that can induce cancer by integrating their viral DNA into the host cell genome in a way that disrupts the normal cell regulation. This can lead to uncontrolled cell growth, a hallmark of cancer. The integration can activate the oncogenes, that are the cancer causing genes, or inactivate the tumor suppressor gene, triggering the imbalance, and it can cause the development of cancer. There are certain examples. The first one is human T leukemia cell virus, also called as HTLV. This retrovirus is linked to adult T cell leukemia or ATLV W12L, a type of cancer that affects the white blood cells. The second example is avian leukosis virus, also called as ALV. It is found in birds. This virus can cause a variety of tumors including the lymphomas and sarcomas in birds. The next category is lentiviruses. Lentiviruses are another important group of retroviruses known for causing the slow progressive diseases. Let us explore the characteristics and types of infection they cause. Lentiviruses are retroviruses that cause very long term and chronic infection. The name lenti means slow, which reflects the slow progression of disease these viruses can cause. They are characterized by prolonged incubation period, meaning the symptoms may not appear until a year after the infection has occurs. There are some examples. The first one is HIV, human immunodeficiency virus. The most well known lentivirus. HIV causes AIDS, that is acquired immunodeficiency syndrome. HIV targets the human immune system, gradually weakening it and leading to life threatening infections and various cancers. The second one is simian immunodeficiency virus, also called as SIV. This virus infects the primates and it is closely related to HIV. It is believed that HIV originated from SIV after crossing into the humans. The next category is spuma viruses. The third group, spuma viruses, are less known but still important. So, spuma virus, also known as foamy appearance, the spuma virus, also known as foamy viruses, are unique class of retroviruses. They are named after a foamy appearances that they cause during the infection. Unlike oncoviruses and lentiviruses, spuma virus are generally not associated with disease in their natural hosts and they tends to cause asymptomatic infections. So, till now we have looked how these retroviruses life cycle occur and how they can infect and cause various types of disease. Now, briefly we will touch the retroviruses and their application in biotechnology. One such application is retrovirus and gene therapy. So, retrovirus have found an important place in the field of gene therapy due to their ability of integrating their genetic material into the host genome. This capability can be harnessed to deliver the therapeutic genes to a particular cell, potentially correcting the genetic disorders. However, while the use of retroviruses in the genetic therapy offers a significant advantages, it also carries certain risks. Let us explore how retroviruses are used in the gene therapy along with their potential risks and challenges. The key to use retroviruses in gene therapy lies in their ability to serve as a vector or carrier for the therapeutic gene. Let us discuss how retroviruses vectors work and why they are useful. In gene therapy, retroviruses are modified into the carrying the therapeutic genes instead of their own viral genes. This modified virus, called as retroviral vectors, are engineered to replication inside the host cell. When these vectors infects a target cell, they integrate the therapeutic gene into the host genome, potentially correcting the genetic disorders by enabling the cells to produce a functional protein that was missing or defective in earlier cases. The ability of retroviruses to integrate into the host genome makes them particularly effective for the long term gene expression. Once the therapeutic gene is integrated, it is replicated along with the cell's DNA every time cells divide, ensuring the correction passes into the daughter cells. Now, let us also discuss some of the risks associated with this process. The main advantage of using retroviruses in gene therapy is their potential to correct the genetic disorders at the source. So, by delivering the functional copies of genes into the cell that have defective or missing version of retrovirus vector can potentially correct the underlying cause of genetic diseases. For example, in conditions like SCID, that is severe combined immunodeficiency, where the immune system is compromised due to genetic defect, the retroviral vector can introduce a normal copy of defective gene, restoring the immune functions. Because retroviral vector integrate their therapeutic gene into the host genome, the correction is potentially long lasting, providing the continuous protection of the needed protein or the life of a particular cell and its descendants. This makes retroviral vector special and valuable for treating the chronic genetic disorders. While the use of retroviral vector offers great promise, there is also a significant risk associated with their ability to integrate into the host genome. This risk, also known as insertional mutagenesis, must be carefully considered. Now, the insertional mutagenesis will occur when the integration of the retroviral vector interrupts the normal functioning of host cell. This potentially leading to the unintended consequences such as activation of oncogene, cancer causing gene, or the inactivation of the human suppressor gene. This disruption can lead to the development of cancers or other serious conditions such as normal regulation of cell functions and divisions. The random nature of integration means that the therapeutic genes could potentially be inserted into the harmful location in the genome. While the chances of this happening are relatively low, the consequences can be severe, making it a major concern in the development and application of retroviral gene therapy. So, with this, let us conclude our today's lecture. As we have explored throughout this presentation, retroviruses are more than just agents of disease, they are also very powerful tools in biotechnology and in the field of medicine. Their unique ability to integrate into the host genome has provided both challenges as well as opportunities from understanding how they can cause the diseases like cancers and AIDS, to harness their potential in gene therapy and vaccination development. The key takeaways from today's lectures were understanding the retroviruses, impact on the human health, and its application in the field of biotechnology. So, with this, let us end today's lecture. Thank you very much.